Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond. Welcome back to another Tools for Looking into the Brain episode, part of our mini-series in collaboration with Scientifica, where we explore neuroscience methods and techniques and the world of academia. Today we are joined by Professor Dave McLean, a principal investigator and professor of neural development and behaviour at the University of Edinburgh. Dave obtained a BSc in biology, followed by a PhD in neurobiology with Keith Sillar at the University of St Andrews in Scotland, where he studied the mechanisms underlying motor behaviour in developing frogs. Following that, he carried out a postdoc with Joseph Fecho at Stony Brook, then Cornell University, studying motor control of swimming by spinal cord neurons in larval zebrafish. In 2008, he became an assistant professor of neurobiology at Northwestern University before moving to his current position at the University of Edinburgh as the chair of neural development and behaviour in 2025. His lab is interested in how neural circuits in the brainstem and spinal cord develop and ultimately control the speed and direction of locomotor movements in developing zebrafish by using a combination of behavioral, imaging, electrophysiological, and molecular approaches. I'm looking forward to hearing more about your research today. Welcome to the podcast, Dave.
Well, it's great to be here. Thanks for the invite.
So let's start as we normally do. I just want to ask, how did you first get into neuroscience and into this specific topic of neural circuits of locomotion?
When I was looking for colleges, I was really thought of myself as more of an artist, but thought, the practical aspect of getting a job meant science might be a better idea. I didn't realize that you could also be a starving scientist, but that's for probably another podcast. And so, yeah, I was born in Scotland, and my parents went to University of St Andrews, and so I was really keen on going back there to study and chose environmental geoscience. I thought that would be a pretty surefire way to get a job. And as part of the course, we had to take a lot of biology courses, and just the information there was just so much more exciting to me than studying rocks. And so I shifted to biology, and I was really interested in human physiology, but also animal behavior. So watched a lot of nature programs, like David Attenborough, Dan Fossey, all these fantastic scientists who are amazing communicators and really got me excited about animal behavior. And so, in the sort of synthesis between physiology and behavior in neuroscience, the physiological basis of behavior seemed like a natural topic. And I was really lucky at St Andrews to have some amazing professors who were studying simple model organisms and motor circuits in particular. And so I managed to hook up with Keith Siller as an undergrad, and yeah, stayed on to do my PhD, and then just continued on from there. And so that was really what got me into the study of circuits and locomotion, was just my experiences as an undergrad, and the fortune of having some amazing mentors.
That's really nice. And I guess, yeah, motor control is like the most important and basic form of any behavior. So
Yeah, it's pretty fundamental. I mean, the other advantage is that there's a really an overt representation of the circuits at work, like you move. And it's also rhythmic, and so a lot of circuits in the brain operate as rhythm generators. So trying to understand the basis of how cells and synapses and circuits come together to generate different frequencies and phases and amplitudes of rhythm, motor behaviors and locomotion in particular is a really good model for trying to get at that. So
And so I know that you now currently study like motor control in zebrafish. Can you tell us a bit about why zebrafish are a good model organism for this? And also, how do you actually study that since they're, swimming?
yeah, so that again, there's like a boring backstory to that. I mean, for my PhD, I was interested in neuromodulation. So how different chemicals like 5-HT and dopamine and noradrenaline can change motor outputs. And to test that, we used to just chuck on drugs into the solution and then just see how it affected things. And then in the discussions, we talk about the natural sources of this, like nuclei in the brain that actually release serotonin and dopamine, and how this might be acting in a more naturalistic fashion. And so my PhD advisor Keith had been at a meeting and saw a talk by Joe Feccio, who was doing in vivo imaging of brainstem nuclei as the animal was behaving. And he really pioneered a lot of these approaches using chemical dyes that reported neural activity, calcium indicators, basically, kind of the precursors to the genetically encoded versions. And I just remember being totally fascinated by that. Like, oh, that's what you want to do, right? You want to be able to actually image the nuclei as the animal's behaving and test some of these ideas that we think are happening pharmacologically. And so that's what brought me to Joe's lab. And that really kind of highlights the power of zebrafish, is that they're completely transparent during their initial phase of development. They go from a fertilized egg to a foraging organism in like a week. So it's really rapid. They're see-through. There's been a lot of effort to create genetic tools to leverage, monitoring activity, manipulating activity, looking at structure, function. And so, and particularly if you're interested in development like I am, you have this kind of see-through organism that is developing, becoming mature, its motor repertoire is improving, and you can actually visualize the circuits as they develop. I mean, imagine if you were interested in like the combustion engine and I told you there was a simpler plexiglass version, right? I mean, you'd probably be like, yeah, okay, I'm going to study that. And so that's really one of the powers of the model is its transparency, simplicity, and accessibility, I think.
I read that in some of your studies, you recorded neuron cell attached and you did whole cell recordings while monitoring fictive swimming. Could you tell us a little bit about how this is even possible? So, are the fish moving or are they stationary? Like, how do you do it?
Right. So, I mean, like I just highlighted, one of the advantages is you can do a lot of things in intact fish while they're moving, and you can do head fixed stuff where the tail is free to move. And the problem with the spinal cord and with electrophysiology, as you point out, is that it's not very amenable to even small movements, right? So what we do is we use chemicals that block the neuromuscular junctions so that they can't actually move. So they're fixed to a cell guard surface, and that allows us to use suction electrodes to monitor the peripheral motor nerve activity that would normally drive the repetitive swimming commands. Because the neuromuscular junction is blocked, it won't actually move. And so that is an approach I learned as a graduate student in tadpoles, where you can throw alpha bungarotoxin on the tadpoles, and that will block. And there had been decades of work pioneered by Alan Roberts and his colleagues kind of studying spinal circuits using these so-called fictive preparations. And so that sort of immobility provides the stability you need to do the patch recording. Now, zebrafish are like half the size of the tadpoles, which was a shock to me when I first started moving them. So you do need high-powered microscope and motorized manipulators to get your electrodes in there, and pretty steady hands to do the dissections. But the information you can glean from whole cell recordings in terms of cellular properties and synaptic dynamics is much higher temporal resolution and very, very super informative compared to the slower approaches like calcium imaging, where you really don't have that sort of resolution. And it's usually limited to, say, super threshold, spiking type activities. So you can get at subthreshold synaptic dynamics using the electrophysiology as well.
That's fascinating. Both Clara and I have done patching. I mean, Clara still does it on her day to day. And we know how challenging it is
Yeah.
to even patch on slice, let alone a whole cell whilst the animal is like awake.
Yeah, yeah, it's super amazing. Um,
Yeah, yeah, and this is great. I mean, the great thing about studying the stages we do as well is that they're completely intact. So their visual system, their auditory system, their somatosensory system. So we can really not only use more naturalistic stimuli to trigger different outputs, but also explore the full range of their locomotor repertoire. Normally to provide access for these types of recordings, you have to do some pretty heavy dissections, pull the nervous system out or do slices, like you point out. And so that again, I think is a huge advantage. Their size and their relative youth, being less than 5 days old, and they're already capturing prey and avoiding being eaten. So it's they're super precocious in that sense.
And am I right in thinking that the fish would be like embedded in agarose or some medium?
Yeah, so for the imaging, we that's exactly how they would be restrained. And but for this, we actually use finely etched tungsten pins to secure them to a sill guard surface. So kind of like with a slice, I guess you could use mesh. But
Oh,
yeah, and again, that was something that was originally pioneered in tadpoles that I kind of moved over to zebrafish when I joined Joe's lab as a postdoc.
I was also wondering, so of course that's amazing that you can gain insights into the neural activity during motor control, but if the fish are not actually moving, this is obviously different from like normal movement where they would also be receiving visual information of
Yeah,
changing surroundings. And so I
yeah.
was wondering, do you also use this in combination with like some sort of virtual reality or some other like visual stimuli, or do you just like exclude that in your interpretation of the data or
Yeah, so a lot of what we are interested in understanding are so-called central pattern generators. So these are circuits that, in the absence of sensory input, can generate appropriate rhythmic activity to pattern your movements appropriately, whether it's fast or slow or changing direction. But of course, what we're also interested in is more realistic kind of stimuli, and what we've been exploring more recently is using visual stimuli to mimic looming like predators or small moving dots that would mimic prey. And we've done a lot of behavioral characterization of real-time interactions between predators and zebrafish and their prey. So we have a good sense of the types of movements they'll produce and what those should look like in more kind of fictive conditions so that we can try and replicate that and really start to explore the cellular and synaptic mechanisms that drive it. But yeah, absolutely. Most of these stimulations are ballistic, movements where the fish is trying to escape. In cases where they're chasing prey, you want to differentiate between so-called open loop and closed loop type stimuli, right? So most of what we do is open loop. Closed loop, you can use the motor nerve recordings, and this is something that was pioneered by Misha Ahrens and Ruben Portugues in Florian Engert's lab. But you can use the peripheral nerve recordings to update the visual scene so that the fish actually it changes in response to whatever the fish is doing, whether it's turning or swimming quickly or swimming slowly. And they've done some super cool experiments where they can uncouple the visual stimuli from their movements. So it moves slower or faster than it's expecting. And then the fish kind of becomes startled and changes its behavior quite rapidly. And so, yeah, those are really powerful approaches that are, again, possible in zebrafish because of their accessibility and the fact that you can do the experiments in intact conditions.
I have a follow-up question related to this. So I also noticed that you've studied the cerebellum and its
Yeah,
output neurons, and I'm also researching the
yeah,
cerebellum. And so much about the cerebellar literature is about how the internal model gets updated based on the movement output, and it's this loop. And I understand that you have these other techniques to alter the visual stimuli and things like that. But what about when you require like physical feedback to be able to then execute like a sequence of movement? You see that a lot in disease models with like dyspraxia or Parkinson's, where you see a lot of these movement deficits due to the inability to update the model and to regulate certain things. So how does recording this activity in a paradigm where the zebrafish
yeah.
is fixed and you're removing from the equation that feedback system?
Yeah, yeah, that's super important. And and that it really depends on the sort of question you're trying to ask in relation to cerebellar circuitry. And again, I would defer to cerebellar experts in this sense. And again, Ruben Portuguese is someone who's been studying this in really with some very beautiful experiments, trying to dissect internal models of, an expectation versus, the external world and the outcome. And that is clearly one of the major functions of the cerebellum, is to kind of compare self from other and your performance. And so in that sense, I think sensory input's critical. But there are different senses that it processes, right? And so we can't. You could imagine devising experiments where you're changing flow of the water, like with little pico spritzers or something. But the good thing about zebrafish is they're highly visual creatures. If you see a picture of them, their eyes are huge compared to the rest of their body. It'd be like you and I holding like two beach balls on our shoulder. And so a lot of the information they gather from the world is visual. And so if you have control of their visual world, which is relatively straightforward, then you can provide that sort of match between internal models and external stimuli and start to understand how the cerebellum is encoding that. Other models that are probably better at that, like electric fish. I don't know if you're familiar of any of Nate Sawtell's work in electric fish at Columbia, but he does some really, they can take advantage of the fact that they sense using electric pulses from modified muscle cells. So their actual sensing ability is a motor command. It would be like if the retina had LEDs that was flashing light, and then you were trying to decode that as it bounced back. And so in that sense, they can do some really accurate analyses of what the internal model would be versus expectation, and it's all really beautiful. The work we did in the cerebellum was much simpler. We did kind of a classic Pavlovian type experiment, where a relatively innocuous visual stimulus, if you paired that with a tail shock, kind of like eye blink conditioning, they started to respond to that stimulus. And not all of them did. Like 50% of them were non-responders. So there's quite a lot of variability. But you do see this acquisition of an association. And this happened in minutes, like tens of minutes, which is totally in the realm where you can hold a recording. So it was kind of cool. If you think about eye blink conditioning, this is multiple trials over many days. And imagine trying to record a patch, hold that the entire time. It's impossible. But we could do it in zebrafish. And so we're able to monitor changes in the synaptic dynamics within the Purkinje cells and their major output neurons, which are called eurodendroid cells in zebrafish, but that would be like the deep cerebellum nuclei, essentially. Some really talented PhD student and postdoc were able to do these recordings and really look at how the dynamics within the cerebellum were changing as it learned a new task, which I think is still pretty cool, even if you're not controlling the sensory environment, right?
Yeah, very cool. I found it fascinating how you were able to adapt that Pavlovian conditioning to zebrafish. That was, yeah.
Yeah, that was
And
fun. That was really thanks to Indira Raman, who is a cerebellar neurophysiologist who had really kind of deep understanding of all that circuitry. So that was a really fun collaboration for me.
yeah, kind of along similar lines. So you mentioned this concept of a central pattern generator, which I feel I've like heard about and learned about on several occasions, but I still always feel like I don't fully understand it. And so I was firstly hoping that you could explain this a little bit, but also I was wondering like what is the role of the spinal cord versus the brain? Does this sort of rhythmic movement is that purely coordinated by the spinal cord, or is there also feedback from the brain?
Yeah, that's a great question. How much time do we have again? I can get into this in quite a lot. Okay, so yeah, so the idea of a central pattern generator, as I said, is that you don't need sensory input, you don't need patterned input from the brain. You can essentially just have a go signal from the brain. I need to walk, and that can be completely unpatterned, just like a tonic input. Imagine just dumping glutamate onto the spinal cord, and then you get this beautiful rhythmic output. And so you probably all heard like a chicken with its head cut off. Anecdotally know that the spinal cord is totally capable of doing this on its own. And there were some experiments actually in the turn of the 20th century by Thomas Graham Brown, who was a student of Sherrington's. And at the time, they really thought that locomotion was generated as a chain of reflexes. So you actually move through the world by interacting with it, right? Your foot would hit the ground, that would give you some sensory input, which would cause your other leg to swing forward, and then that would hit the ground, that would give you sensory inputs that would cause your other leg to swing forward, right? And so he did this deceptively simple experiment where he essentially severed the dorsal roots, which is where all the sensory information enters the spinal cord. And then was able to show by severing the spinal cord, which essentially just was a tonic dump of all the neurotransmitter from those axons that got severed. You got this beautiful alternation, which looked a lot like locomotion. So, the idea that the spinal cord contains the necessary circuitry to generate locomotion has been known for quite a while. What we've been really trying to figure out is, the composition of that, and in particular, how is it that different speeds of movement are regulated. So if you think about us, we can wander, we can skip, jump, run, sprint, walk. Horses can kind of canter, gallop. Cats can bound. As you move more quickly, there are different patterns of coordination involving your limbs and different frequencies and amplitudes of muscle contraction that drive that. And so, how are those distinct kinematic functions distributed throughout the spinal cord? And so, in that sense, the brain is really playing a key role in not only turning things on, but probably matching sensory needs to motor output, right? If you see something chasing you, you're going to want to run fast. If you see something you want to eat, you're going to run fast too. If you're just exploring, you may want to take your time. If you're on ice and all of a sudden things are really slippy, then you probably want to move more slowly, right? So sensory input and brain inputs play a really important role in modulating and shaping locomotor output. But the idea is that the circuits that ultimately execute locomotion are all within spinal cord. It's just we're still trying to figure out exactly how they distribute these different functions. That makes sense.
Yeah, I'm really glad that it's now been explained to me clearly for once. But are there, just out of curiosity, like, is there only one type of central pattern generation in the nervous system? Is it only for movement? Or, oh, could you have it for like breathing or something else more
Oh,
autonomous?
absolutely. Yeah,
Okay.
anything that's rhythmic, you can think of as central pattern generator, right? So breathing, chewing, like mastication. I mean, it's kind of a loaded term because when people think about central pattern generators, they think about all this classic work. But the brain is a central pattern generator, right? We talk about different frequencies like theta, delta, gamma,
Mm.
right? These oscillations and how different kind of more cognitive behavioral functions arrive from synchronizations between different frequencies. And so those are all rhythms, right? And so they have to be generated by something that is rhythmic. The extent to which they rely on internal dynamics in those brain regions to generate rhythms versus afferents, like inputs from other, I think is a major question, even in the cognitive field. And it kind of like reflects a very old question in locomotor control, right? That is it sensory inputs or is it centrally generated? And so usually what happens in these debates is that it's a bit of both, right? You take extremes and then you have to make concessions. And so I think definitely rhythm generation more broadly is a critical function of the brain. The question is to what extent is that a function of internal dynamics and what do you need in terms of inputs from other regions to help shape that and make sure it's appropriate.
Yeah, like I know that Purkinje cells, for example, they have this tonic firing rate that they mainly receive from they produce from simple spikes from climbing fibers and osteofiber input and things like that. So yeah, the idea, I guess I've heard it in several different terms. I've heard it in terms of like tonic firing, but now this other term of the pattern generation. So yeah, I guess
Yeah.
many different brain regions
Or
have.
pacemaking.
Oh yeah.
They also call it pacemaking, right? So like there are neurons in the basal ganglia that are pacemakers. And so that's kind of like some tonic firing rate and that's another question in locomotor control to the extent to which intrinsic cellular properties like voltage gated conductances that can imbue neurons with an autonomous capacity to generate rhythms versus their functional interactions with other neurons are responsible for rhythm generation. So it's like, do you have a conductor in an orchestra that's telling everybody what to do because it has this internal capacity to to generate the right rhythm, or is it more like a jam band where, people are just the music emerges as a function of they're feeding off one another and you get these really groovy tunes.
That
But
is
yeah,
so
yeah,
cool.
pacemaking, central pattern generating, rhythm generating, it's kind of all interesting
Yeah. I
comparison.
love the way that you phrased it, whether it's like an orchestra or like a jazz band, because I guess the interesting part is when sometimes these rhythms do get disrupted, like when Purkinje cells all of a sudden have a complex spike and that disrupts the tonic pattern. And yeah, with the basal ganglia, the cerebellum also projects to the basal ganglia, and I wonder if the tonic firing input to the basal ganglia then disrupts the basal ganglia pacemaking firing. So yeah, it's very cool seeing these different brain regions interact and seeing whether, like you say, whether it's an orchestra or whether it's a jazz band.
Yeah, and I'm sure that there are cases, but I'm wondering also for central pattern generation of simple movement like walking for humans, or, for example, or swimming for fish. Um, are there cases of this being disrupted?
Well, I mean, the most obvious one is spinal cord injury,
Oh, okay, so like,
right?
but it just
So
results
yeah.
in. But I mean, I'm thinking like, is it either like a complete aberration of the ability to move, or is there like cases in which it cannot be as rhythmic?
I think, a lot of SMA or ALS, these typical motor diseases which impact, motor neurons. I think there's a growing appreciation that deficits in interneurons may even predate problems that folks are seeing in motor neurons. So the actual interneurons, excitatory and inhibitory, that regulate motor activity so that you can generate rhythms or even discrete movements, right? You can think about reach and grasp. That's not necessarily a rhythmic behavior. Certainly, it looks like these tauopathies, these so-called kind of accumulations of proteins where they shouldn't, are impacting circuits and their ability to communicate with one another. Demyelination diseases, so that signals aren't getting as quickly to where they should. These are all going to impact how things interact, how rhythms are generated, and ultimately your ability to move. Even if you think about, different types of motor neurons that might be more, sensitive to these diseases, like losing your slow motor neurons preferentially. And so now all you've got are your fast motor neurons. And so your movements become much more kind of ballistic and gross, and you don't have that sort of fine control anymore. Certainly, I think, when you think about motor diseases, we've really focused on the output side, the motor neurons, but there's a lot that we still need to know about what's happening, that might even be predictive at the level of, a synapse or two upstream in the spinal cord.
Yeah, I guess that highlights like how many aspects there are of motor control. And so when I was thinking myself about like motor deficits, I was wondering like, oh, is there something that could specifically target just the rhythmic aspect? But I think this is actually a good natural transition into what I also wanted to ask about, which is just more about like the neurobiology of motor control and the different subtypes of neurons that are involved, or like populations or circuitry. And first, I read your paper on hierarchical control of locomotion by distinct types of spinal interneurons in zebrafish, where it was mentioned that different neuron types can control the timing versus the size of the activity, as you mentioned earlier as well. Could you tell us a bit about how this works?
So, a lot of the early work trying to understand the cellular and synaptic mechanisms of CPGs was carried out in things that swim, like lampreys and tadpoles, and zebrafish are really kind of following on that work. And there's really a lot of beautiful characterization of cellular mechanisms, rhythmogenesis, patterns of connectivity, and these interneurons all talk to one another and talk to motor neurons. So you can think about them as not only regulating, say, the timing of movements via their interactions with other interneurons, but also they need to produce the movements themselves by driving motor neurons and muscle contractions. And so for a long time, this kind of dual function in terms of timing and output, right, was thought to be carried out by the same neurons. But there was more recent work after that in limb control, which was providing evidence that maybe the neurons that are controlling timing are different from the neurons that are controlling output. And the experiments, probably the best evidence for that is they would record, say, this fictive motor activity from a particular motor nerve, and then a burst would drop out. So there'd be like no output at all, but then it would come back at exactly the right time that you would expect, right? If it hadn't dropped out, right? There was no kind of reset, right? So these are called non-resetting deletions. And the idea is that if the same interneurons that are responsible for output all of a sudden dropped out, right, and couldn't drive the motor neurons anymore, how in the world are you still maintaining that kind of clock of patterns such that when it comes back the next cycle, it's in the appropriate phase? And so, but the problem is it's really much more challenging to do this sorts of cellular and synaptic assessments. I mean, looking for pairs of connections, recording from neurons in intact cats, for example, or even mice. And so a lot of the inference on kind of a segregation between timing, which is like a network of just interneurons talking to another, and then they talk to other interneurons, which ultimately hand that information off to motor neurons. A lot of that is done through genetic manipulations and modeling. And so zebrafish were a perfect place to kind of test these two ideas because they're fish, right? So they should swim like lampreys and tadpoles. But we had the genetic accessibility that you have in mice, which allowed us to map specifically the populations that would be carrying out kind of homologous functions in mice. Because it turns out, our spinal cords are built exactly the same way using the same genetic programs. So you can really draw pretty clear comparisons between the same types of cells in fish and frogs and mice and humans now. And so, what we saw was actually a kind of a hybrid organization. And so you did have interneurons that seemed to care more about timing, and some that cared more about output or intensity. But they were defined not by like an exclusive anatomical hierarchy, but more by some just had stronger connections to interneurons and weaker connections to motor neurons, and others had stronger connections to motor neurons and weaker connections to interneurons. And so what that means is that you can actually both segregate and integrate timing information. These two layers aren't like a multi-layer. It's like a weighted monolayer. And so you can separate control of the intensity of a movement versus its rhythmicity via this kind of more hybrid model. And so that I think is cool because it really helps diversify motor output. You can independently, you know, regulate the intensity of movement. Say you want to step over something, but you don't want to like change your cadence, right? You're still kind of jogging and then you lift your leg up. If you're driving a rhythm generator to both increase the amplitude of your leg, then you're going to get a frequency change as well, right? You're going to go faster, which might alter your step timing. So it's really critical that you can separately control these two features, how large your movement is versus how fast it is, right? And so this was as a child of divorce, it was very satisfying to be able to say, no, no, you're both right. It's, there are interneurons that are connected to everything, but it's just a weighting scheme where some are having more of an influence than others, right?
I actually just wanted to ask a follow-up or like some clarification because
Yeah.
I think you explained it really clearly, but I just want to make sure I fully understand this whole idea of the circuitry for the timing versus size of the movement.
Mm,
So, as far as I understand, the main determining factor of that makes these neurons either contribute towards timing or size is their connectivity, like what inputs and outputs they have, right?
hmm
So I was picturing it all like in a circuit, and within the same circuit, there are neurons that are either more connected to other interneurons or more connected to motor neurons. Um, and based on that, they have different effects. But how is this then, how does this ensure that timing and size are actually dissociable, because if you like manipulate the activity of one of the subpopulations, does it not also alter the activity of the other subpopulation?
Yeah, that's a great question. And so the one population we were studying, which kind of gets to this point, is a population of interneurons called the V2A interneurons. These are identified by a transcription factor called CHUX10, which labels neurons that are glutamatergic and ipsilaterally projecting in fish and frogs and mice and likely humans as well. And so it turns out the motor neuron ones, they don't talk to one another at all. So I was kind of a little short. I didn't want to get into the weeds of this too much, but the ones that are really regulating motor neuron activity don't have so-called recurrent connections. So if you drive that interneuron, then it's going to primarily drive motor neuron output, um, and have relatively little, if any, influence on interneurons. The connections they make to motor neurons are very reliable, super reliable. So almost 0% failure rates. But the connections that they make to interneurons are much less reliable as well. And so you could imagine that this is like a dedicated path for causing muscle contraction. And so if you want to generate, say, a discrete movement, like a reach and grasp or just like a turn, the brain could fire those, innervate those cells, cause them to fire, and then you just get this one movement, right? Um, Now the problem with locomotion is that you have like a rhythmic input, right? And so in that case, the rhythmic V2As, the ones that are more strongly innervating other interneurons, have again very low failure rates to other interneurons and very weak connections to motor neurons, which are subject to failure on a cycle by cycle basis. But they feed into this V2A motor neuron population. So you can think about the V2A motor neuron population as the nexus for rhythmic inputs for locomotion or discrete inputs, right? And by driving that population discretely, you don't then engage this rhythmic output. But the rhythmic interneurons can co-opt that to generate rhythmic motor activity. And so
Yes.
the fact that they have kind of weaker, higher failure rate connections between one another means that they are talking to one another. And you could imagine that maybe, say, neuromodulators or something could tweak those connections and generate completely different outputs, right? So they're not kind of strictly hierarchical in the sense that you have timing and then intensity, right? There's kind of some mutual interaction, more like a Zen kind of symbol, right? Where they are kind of talking to one another, and you can tweak the strength of that, but you can just get either a discrete movement or a rhythmic movement thanks to the lack of communication between the neurons that are talking to motor neurons. So that's really a dedicated channel to motor neuron output.
It's a good example of how well, I guess how complex neural circuitry is. It's not just like one circuit does this, one circuit does this, but it has to do also with the intricacies of it, and like and things beyond just, yeah, like beyond connectivity, like beyond structure and beyond function, but also like strength or probabilities. Yeah.
Yeah, exactly. And that's something, I think, in the textbooks, we think of motor systems as being very hierarchically organized, right? Everything kind of feeds down through cortex, right? Starts there, and then it gets down to the midbrain and then to the brainstem and then to the spinal cord. But as we're getting better at labeling neurons, particularly with viral approaches, we're seeing a lot of loops, right? Things are talking to one another. It's not just exclusively hierarchy. Things are feeding back, connections are going all over the place. And of course, one problem with viral methods is you can't tell the strength of that connection. You know it exists, but there's probably a lot of subtleties in terms of the relative weights of synaptic inputs, how strong they are, what they're, we're seeing a lot of, say, electrical synapses, these gap junction type synapses that people always thought were exclusively in early development, but now, you see them in a variety of brain regions where you really need to process rhythmic activity at speed. And so those are going to complicate assessments of cellular properties and connectivity and so I think you're absolutely right. There's a lot more complexity there that we're only just starting to tease apart thanks to more modern methods of assessing these, but still a lot to learn.
Yeah, and also like with viral methods or transgenic methods, like you're still confined to targeting cell populations based on genetic expression, whereas you could also define cell populations based on other things like you've mentioned, like connectivity or structure or strength of their synapses or location of their synapses, like synaptic targeting, etc. So yeah.
Yeah, exactly. Yeah, I mean, and we're seeing, I mean, in the spinal cord, there's a ridiculous amount of diversity of interneuron types. Like, it's like, if you think about a motor neuron, there are like 30 classes of interneurons that impinge on that, which is much more even than in cortex, right? And so trying to understand why you would need that sort of diversity is a major problem in the locomotor field. And at least in zebrafish, what we've seen is that some of that diversity is exactly as you say. It's due to the location that synapses are being formed on neurons, in addition to, their target. So I think when trying to explain neural diversity, we have to think not only about who they're talking to, but where and what that functional impact will have on information flow.
Yeah, that's very relatable actually, because like my research topic is about a subpopulation of interneurons that specifically target the axon initial segment. This is in the cortex
The
of the
chandelier
chandelier
cells.
cells. Yeah.
Yeah, 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 yeah.
So, like the one key feature that defines them is their subcellular targeting. So that's what I was also thinking of.
Yeah, yeah. I mean, then you have basket cells and Martinoli cells out at apical tufts. And so in cortex, they've known for ages that there's a really clear link between subcellular targeting and the impact that, inhibitory interneurons are going to have on information flow along a pyramidal neuron, right? With the chandelier cells just being like an ultimate veto, like nothing's coming through. And then you get much more subtle modulation out in the apical tufts. And so I was inspired a lot by that work and pretty amazed that we see the same thing in a fish spinal cord, right? So it seems to be a very old feature of circuit organizations going back probably to, the evolution of the nervous system itself, that you want to really have this kind of control and information flow through your primary output neurons, which in our case are motor neurons, right?
Are there cells that specifically target the axon initial segment in the spinal cord of fish?
Yeah, yeah, there are. Yeah.
Cool.
Yeah. Yeah, yeah. And it's related to development like in cortex and also function, which, of course, as I was saying earlier, we have a much better idea of what these circuits are supposed to be doing because you see movements as a consequence of their output, right? It's hard to kind of assess fictive thinking in a cortical slice, but we can do fictive locomotion, right? In a spinal cord. So yeah, these neurons are innervate the axon initial segment of motor neurons are among the first to develop. And they make sure that when the fish are generating a startle response, so if there's something threatening coming at them, they generate this very rapid, forceful bend away. So they make sure that they cross over and make sure that nothing comes out on the other side that would conflict with this appropriate kind of aversive response.
Wow, that's
And
really
then and then it,
interesting.
yeah. And then it masks with speed. So as you move out to the dendrites, then you're getting interneurons that are active during much more subtle kind of prey capture type behaviors where they need much more precision and variable output. So there's a real kind of functional developmental topography related to veto power in spinal motor systems of zebrafish, which I would expect once people look it's going to be there in mammalian spinal cord as well.
Well, and that's actually surprisingly like logical and makes sense because it does not work like that in the mouse cortex at least. Like chandelier cells are actually the last to develop and they
Yeah,
actually don't have strong modulatory output. So we just don't really know what they're doing. Whereas it sounds like in the spinal cord that makes sense. So that's
yeah,
that's
yeah.
nice to hear.
Yeah, that makes a lot of sense. It's also, there are a couple of key differences. These are glycine is a neurotransmitter in the spinal cord, not GABA.
Right. Yeah.
And these are commissural pathways too. And so chandelier cells are obviously local
Yeah.
interneurons. And if I remember correctly, there's some debate about whether or not they are actually excitatory versus inhibitory as well, depending on the timing at which inputs arrive and their conductances. So yeah, I think some important differences that might be due to transmitter phenotype, regionality, and maybe even like decoupling their output from actual movements, where you really need to make sure if you're doing like one movement, you want to make sure, the other side is not active. So,
Hmm.
but broadly, I think it's pretty cool that you can make these types of comparisons.
Yeah, sorry
Yeah.
for derailing the conversation a bit, but this is actually the first time I ever ta like mentioned my own research on Neuroverse. It's really exciting for me. Yeah.
That's cool. So
No, yeah, I
cool.
was just gonna say it's really cool the anatomical specificity with the behavior in terms of like whether it's like aversive, whether it's running away, or perhaps more timely, well thought out behavior in terms of trying to catch a prey or something.
And just also, I should have asked this in the beginning, but like in terms of the interneurons that you mentioned in the spinal cord, what are they excitatory and inhibitory? Like, are they very diverse in terms of their neurotransmitters?
Right. It's yeah. So it's primarily glutamate and glycine that are the major neurotransmitters that shape excitation and inhibition, respectively. There are GABAergic interneurons and cholinergic interneurons, which are thought to be more modulatory, at least in terms of kind of shaping recruitment patterns as opposed to timing them. But yeah, those are the major ones. And so it's kind of interesting. It's not clear to me why there is this clear dichotomy between sources of inhibition, whether you're talking about cortical versus kind of brainstem spinal cord circuits, where GABA seems to be the dominant inhibitory neurotransmitter in so-called higher brain centers, whereas in the brainstem and spinal cord, it's really glycine. And maybe it has something to do with this ability of GABA to kind of be depolarizing versus hyperpolarizing, and you can modulate things. But yeah, it's those are the major neurotransmitters, and there are canonical subsets that use these but have different projection patterns, right? So some cross over to the other side and make sure you're controlling both sides. Some descend, and then there's some that ascend, descend. So you have a whole kind of parts list of interneuron types that can generate both local and longitudinal coordination, both excitatory and inhibitory. And we're still trying to figure out a lot of the functional implications of that sort of diversity.
Hmm. Yeah, I can
Mhm.
imagine it's a really complex circuitry. And are there also like differences along the like anterior posterior axis? Yeah.
Yeah, yeah. So I mean, in us, we can think pretty clearly about regionalization, right, of the spinal cord with both cervical and lumbar enlargements that help kind of regulate limb control. And it's more subtle in zebrafish, at least at the stages that we study. There's a clear rostrocaudal difference, but it's more of a continuum. As they get older, then they start to develop fins. So they have like paired fins, like pectoral and pelvic, which are thought to be analogous to our limbs. But they also have medial fins, so a dorsal fin and a caudal fin that um, that can provide some sort of stability as they're moving through the water. And then in those regions, you do see kind of like a slight swelling, so an enlargement, suggesting that the addition of fin control um, provides an additional burden on circuit complexity, where you need more neurons and more connections to really provide that. And there's some really beautiful work carried out by Jeremy Dason and David Shopik studying the skate. I don't know if you've seen that fish, but
Oh
they
yeah.
have they have like little limbs that they kind of walk around the ground on, and they
Oh,
were
I've
able to
seen
show,
that.
yeah, they
Is it like the
show that
tripod fish?
it's not the tripod fish, but it's
Uh,
similar. Yeah, similar. It's this kind of basic idea where a lot of the Hox genes that ultimately specify limb motor neurons are totally conserved in these fish. So there's like this idea of fins being the precursors to limb has a lot of good scientific evidence for sure. So I think it's an important question, like understanding how the trunk and the limbs interact to generate locomotion.
Yeah, wow. I have so many questions actually. Like now I'm just thinking about octopuses, and then I was like, wait, does it have
Oh
a spinal
yeah.
cord? And like, where's the
Oh
spinal cord?
yeah. No, octopus are amazing. They have a totally decentralized nervous system, so all of the limbs are effectively autonomous,
That's so
Right, yeah,
cool.
that makes sense.
right? But there's yeah, but there's read this book called Other Minds, and I forget the author, but it's fascinating talking about
I have
intelligence
that
of
book. Yeah.
octopus. But anyway, but it's interesting because there seem to be like biomechanical problems, even if you're a soft-bodied animal. When octopi, plural of octopus, when they reach for things, they actually generate kind of articulations in their limb, a lot like joints,
Mm.
so that they can control things. And there's a group in Israel that studied this and demonstrated that the basic biomechanics of reach and grasp are same whether or not you've got a floppy arm or a rigid one like us with joint articulation. But yeah, the idea of how the limbs are kind of like their own little fiefdoms is fascinating given how good they are at moving, right?
Yeah.
So I
Yeah.
think that's a really cool question.
So on that very unrelated but very interesting note, I think
Yeah.
we'll wrap up the episode for today. So thank you again so much for joining us, and it was really interesting hearing about your research and just generally the spinal control of locomotion.
All right. Well, thanks for having me. This was a lot of fun.
Yeah, thank you, David. It was super fun. Looking forward to seeing how your lab does in Edinburgh.
Yeah.
All right. Thanks so much. Let
Okay.
me know when you're up here.
Yeah, will do.
Yeah. Thank you for listening to this episode, and we hope you enjoyed it as well. If you're interested in learning more about Neuroverse or checking out our Tools for Looking into the Brain series, you can find information on our website, neuroversepod.com, or follow us on social media at neuroverse_pod. And thank you again for Scientifica for sponsoring this episode. To learn more about electrophysiology, or if you want to set up your own electrophysiological systems, check out Scientifica's website, scientifica.uk.com.